Okay, one more video on enzyme kinetics, and in this case, we're going to look at a few examples to really drive home the meaning and importance of this Michaelis constant and the kcat. So let's look at some examples right here. Notice that we have our constant values, and we have this ratio right here. Okay, this ratio is our catalytic efficiency. Let's think about why we would care about that. So we said that low values of Km meant higher affinity of the enzyme for the substrate. So lower values meant it was more likely to bond to the substrate. Okay, right here, this was our turnover number. So if you remember, that was how fast the reaction proceeded once it was bound. So let's say reaction speed once it was bound in that substrate. Okay, so ideally for an enzyme, you'd have a low Km and a high kcat. And so this catalytic efficiency number talks about both of those, right? The higher this number, since it's kcat on top, low Km on the bottom, the higher the number, the higher the catalytic efficiency. So it's talking about the overall ability of the enzyme to convert substrate to products due to both the binding efficiency. Okay, that's this one right here. We could say that, talk about that as binding efficiency. And due to the speed of the reaction once it's bound. All right, so we've got kcat over Km. The kind of maximum value you can get here is about 10 to the 8th or 10 to the 9th. Okay, we call that the diffusion limit. Essentially, that is the limit based on how fast the electrons will move to break bonds when it's reacting for the kcat value, based on how long it takes the substrate to diffuse through the solution to reach the enzyme. So that's kind of our high end, is this diffusion limit right here. So if we saw a problem like this, use data provided to determine whether the reactions catalyzed by enzymes are diffusion controlled. That's what we mean. We mean, is the limit to this simply the fact that the movement of the electrons, the atoms, the bonds, all of that stuff? Okay, are we near that diffusion limit? If I look at this, right, this middle one has the smallest number here. So this one has the highest affinity, binding affinity. But then I look over here and wow, this one's going to go a lot faster once it's bound. And if you work this out, I believe you get around 10 to the negative 4th here. Oh, nope, that doesn't seem right. 10 to the 4th. Okay, right here, I think you get about 10 to the 8th and about 10 to the 9th. So these two would be the ones that we'd say, wow, those are going pretty much as fast as they can. Also want to look at the importance of some of the individual numbers. Okay, so when we look right here, let's look at the carbonic anhydrase, and let's look at the lysozyme. If I look at carbonic anhydrase, it has a really high turnover number. It reacts really fast. It's got a pretty high, low affinity or low binding affinity, but because it's a pretty high value of that Michaelis constant. But let's think about what this does. Okay, so we need this carbonic anhydrase to convert carbon dioxide to carbonic acid. There is a pretty high concentration of CO2 in the blood, so we don't need a super high affinity. There is so much carbon dioxide there that it's going to bind. But what we do need is we need it for it to bind fast enough so once it's bound, it'll react fast enough to keep our blood pH neutral, to keep that reaction going. On the other hand, if you look at lysozyme, wow, it is pretty slow compared to the carbonic anhydrase. But wow, it's got a much lower Km, a much higher affinity. Okay, so if you think about what this does, this is actually an enzyme that helps degrade bacterial cell walls. This really low Km value means that it's going to essentially seek out bacterial invaders in your blood at really low concentrations, and it's going to bind to them. And once it's bound, it's okay that it's reacting a little slower. Okay, but what we needed was for it to find those things and bind. So both the kcat and the Km values are important.